Welcome back, life science learners, to our final installment. We're looking at protein synthesis today, and in this final segment, we're looking at gene coding. What does it mean? Have you ever wondered about genetic coding? Well, essentially, the process of genetic coding is universal. What do I mean by that? So, let's try and unpack that. If DNA is universal to every organism, but it's unique to the way it is arranged, the process of protein synthesis has a similar mechanism. However, the arrangement of those amino acids may differ. Let's try and understand what do we mean by genetic coding or gene coding. Right, so the genetic code of an organism is a set of rules used by cells to translate information which are encoded in genetic material, which is DNA or mRNA sequences, which are made up of triplets, and that is making proteins. What is important is that in genetic coding, we refer to a genetic coding table. This points to a full set of relationships between codons, which are found on the mRNA, and amino acids. And this is called the universal genetic code. And so that genetic code is often summarized in the table on the next page. I'll show you that in a little while. So the genetic code table is a full set of relationships between codons and amino acids. And this is, as I mentioned, is often summarized in a table that is universally used. Let's look at this. So here is the arrangement of all possible letters. If you look at this, we have 64 possibilities of arranging the bases A, G, C, and U. And if you look at it, we've got various possibilities. So this is a standard universal genetic coding table. It is made up of all of the mRNA codons that possibly may exist using 3 possible letters. So if you look at it, you've got in this box, you've got UUU, which essentially points to a possible codon that codes for an amino acid called phenylalanine. And so this consists of the various different codons that code for the possible amino acids that we have. There are 20 different amino acids, all of which are coded for by this table. What you would notice that in some instances, we have several codons coding for a single amino acid. And in other instances, we might have a single amino acid coded for by a single codon. So this codon table is unique, and that it is also universal to every species that has the process that carries out the process of protein synthesis. What is important for you to be able to do is be able to read the amino acid codon table. What do I mean by that? So let's try and understand that. So we've noticed that there are many amino acids that are present on this table. So this means that if we look at the amino acid leucine, and that is right here, you will notice that the mRNA for that amino acid leucine, I'm going to quickly find that on here, is made up of 2 possible codons, UUA and UUG. As I mentioned, that these 2 codons code for the amino acid leucine. An important point is that the genetic code, as I mentioned, is universal. What do I mean by that? There are some exceptions to this, but this is a process that is similar in every species, where an amino acid would be essentially the same in all organisms coded for having the same codon. So if we unpack this sequence, let's try and understand how do we get the amino acid sequence from this. So let's try this out. So this is a sequence of DNA, or it's one strand of the DNA that codes for the gene. We need to convert this DNA into the mRNA before we figure out how to code that gene. So what would be the codon for, or the mRNA for this sequence? We can unpack that. So TAC, so you're going to have T base pairs with A base pairs with U, C with G, and if we go with C, it's G, C, U, T base pairs with adenine, adenine base pairs with uracil, and cytosine base pairs with guanine. So what we've picked up is that we've got a set of 3 codons, and these are the codons that we now refer to as the mRNA codons. And so this can now be put into a table where we can figure out which is that amino acid that is configured by or coded for by AUG. How do we read AUG? So if we have AUG, A points to the first base, so we've got to look in this specific column, and that's A, so we've identified A. Uracil is the second base, so we use this row and we look for uracil in that. Okay, so it now means that we've got to be looking in this box. And the third base is G, and if we look around the third base, that's where we read that from. And so if we look at G is there, and if we go across, come down that line, we can see that AUG is present there. And in this case, AUG codes for methionine, and it's represented as an abbreviation, which is often referred to as the start codon. Let's try doing that with the second codon. And the second codon is GCU, and if we refer to GCU, again, first base G, so we're going to look here. Second base C, so we're looking in this column. And the third base is uracil, so it's going to be in this box. We've got to find GCU, and GCU is that specific codon. And if we look at that codon, it codes for the amino acid called alanine. And so you often worry whether you need to know about the names of these amino acids. Actually, you don't, because these will be given to you in a table. Okay, so I've got a question or two that we probably can do before we wrap this section up. So here's a question that shows you a process or processes occurring. I'm going to try and attempt a few of these questions before we wrap the segment up. So the first question is, provide labels for structures A, B, and D respectively. So when we look at this diagram, we can see that we've got a series of steps indicating the process of protein synthesis. Let's try and understand what structures A, B, and D represent. So when we look for A is right up here, and that again points to the structure in which all of this is occurring. And so that points to the nucleus or the nuclear membrane. So that's the nucleus, and that's the nuclear membrane that surrounds it. Structure B points to the mRNA, and we can see that the mRNA is produced here. It's now leaving the nucleus to move to the cytoplasm, and so B would be the mRNA molecule. And finally, we've got to look at structure D respectively. And if we look at structure D, guys, we can see that D is a double-stranded molecule, and that is essentially DNA that is coding for the specific gene that we need to produce the protein of. The next question, state one function of molecule D. Now guys, if you go back to DNA and what we're currently looking, DNA codes for life, and if we try and unpack that further, what do we mean by that? It codes for genetic information, and that is often for the production of proteins. And so proteins are produced because we get that information from DNA, and that is due to the expression of the characteristics of the cell. Interesting question. Question C. Which part of protein synthesis takes place at 1? So guys, here is the diagram that illustrates a specific part of protein synthesis. Which part of that takes place? If you recollect, protein synthesis occurs in 2 steps. We've got the first step is transcription, and the second step that occurs outside in the cytoplasm is called translation. And so if we refer to what is happening in that process, so that is the production of mRNA, And we can clearly see that that is mRNA, and that process involved in the production of mRNA is called transcription. Remember, I often use the word script, and the word script should tell you that it's a set of information. So a script contains a message. And so if you think of a message, use that as a reminder to help you remember that that's the production of mRNA. Cool. The next question. Name the type of proteins that control the process named in question C. So guys, as I mentioned earlier on, that this process of transcription involves an enzyme, and that enzyme controls this process here. And that process is controlled by an enzyme called RNA polymerase. And so RNA polymerase is an enzyme that actually carries out the process of transcription. We've got a now identify organelle C, and if we go to this diagram, we can see that the mRNA is leaving the nucleus and the instruction indicates that it's moving to organelle C. Guys, organelle C is where protein synthesis takes place, and if you recollect, it's called the ribosome. And it's often indicated by, as I indicated, the subunit have a structure having two subunits. It's also called rRNA, which is one of the three types of RNA molecules. So it occurs in the ribosome. Let's look at question F. Name and describe. So there's two parts to this question. We've got to firstly name, and second part, describe the part of protein synthesis that takes place in organelle C. And this is a 6-mark question. It's important that we break this up into the number of marks allocated per the question. So the name would be a mark for the name, and describing the process would be 5 marks. And so there would be 5 significant facts that you would need to use to explain this. So the process that involves the protein synthesis that takes place in organelle C, again, we're referring to that process in organelle C as being translation. And so if we go back to the image that we were using earlier on, okay, I've indicated the process of translation occurring here. So we've got to go and remember that we've got to talk about that the mRNA contains codons. Second point would be that the ribosome now reads the codons. It will then instruct a tRNA molecule to bring in an amino acid. You will find that there is a codon and an anticodon, that base pair. Each amino acid that is brought in is connected to the previous one with a bond called a peptide bond. And this process continues where the RNA reads the mRNA, bringing in tRNA molecules with amino acids, and that length of the amino acid chain continues to grow, eventually forming what we refer to as a polypeptide chain, and that is then compacted and configured into a protein molecule. So guys, we've kind of looked at the process of protein synthesis today. We've looked at RNA. We've spent some time understanding the structure of RNA. What is crucial to today's lesson was trying to understand the process of protein synthesis, where it occurs and how it occurs. In this last segment, we've looked at the concept of gene coding and using the mRNA codon table to figure out the sequence of amino acids. As I mentioned, amino acids are put together by the ribosome into a polypeptide chain, which is then configured into a complex protein. All of this happens due to the instructions that come from the DNA. You now understand the process of protein synthesis. You should be able to read an mRNA codon table to be able to synthesize a short sequence of amino acids. Guys, you've done well. You've stayed focused, and you now understand the process. I encourage you to take some questions on this, try and attempt a few questions based on some past papers, and refine your understanding of the process. There's some interesting questions that you can look at. I wish you well. Thank you for staying tuned. See you soon.